Hello everyone. I am Dr. Malarkodi Velraj, Professor and Head, Department of Pharmacognosy, School of Pharmaceutical Science, VISTAS. Today I am going to take you to a fascinating world of plant secondary metabolite alkaloids. To start with alkaloids, we are going to see about the definition, occurrence and distribution of the alkaloidal families, properties, classification, extraction, chemical test, and quinoline alkaloids. Definition of alkaloid. They are a class of compounds which typically contain a nitrogen nucleus in the complex ring structures having a pharmacological effect on humans and animals. Occurrence and distribution. They are distributed in several parts of the plants, like leaves as in case of tea, in barks as in case of cinchona, in roots in Rauwolfia, and in fruits like tomatoes. Properties of alkaloids. They are colorless crystalline solids. Free base alkaloids are soluble in organic solvents. They are basic in nature, insoluble in water. They have sharp melting points, and the alkaloidal salts are soluble in water. They have a very bitter taste. They are partially soluble in organic solvents, and some of the alkaloids are colored, like means of sanguarine in bright red color, berberin in yellow color, and some alkaloids are liquid in nature, like nicotine. And the main property of the alkaloids is to defend plants from insects and animals. Classification of alkaloids. They are true alkaloids, protoalkaloids, and pseudoalkaloids. So coming to see about the true alkaloids, the true alkaloids are organic nitrogen-containing plant compounds where the atom of the part of a heterocyclic ring. They are derived directly from the amino acids and exhibit high biological activity even in low doses, like morphine as in case of opium, cocaine from coca, and quinine from cinchona. Classification of alkaloids. The classification of alkaloids are different types like pyrrole and pyrrolidine. In example is hydrine and coca species. Pyridine and piperidine examples are aricoline, anabasine, etc. Pyrazolidine ring alkaloids, echinamine and seniconine and senisphilin, tropine containing alkaloids like atropine, hyoscine, hyoscyamine, quinoline alkaloids, quinine, quinidine, etc. Isoquinol alkaloids like morphine, codeine, emetine, and cephalin. Protoalkaloids. The protoalkaloids are simple, naturally occurring organic compounds. They contain nitrogen derived from amino acids, but unlike alkaloids, the nitrogen atom is not a part of a heterocyclic ring system and typically feature the nitrogen in the side chain or attached to a carbon skeleton. The examples are ephedrine from ephedra, capsaicin from capsicum, and colchicine from colchicum. Pseudoalkaloids. Pseudoalkaloids are nitrogen-containing organic compounds and the functions like alkaloids but do not originate from the alkaloids from the precursors of amino acids. Instead, the nitrogen atoms come from other building blocks like purines, terpenes, or steroids and an alternative metabolic pathways like tea plants, atropa, and aconitum. Extraction of alkaloids. The powdered material containing alkaloidal salts is moistened with alkaline substances and they combine with acids, tannins, and other phenolic substances to set free the alkaloidal bases. Then the extraction is carried out with petroleum spirit and the concentrated organic liquid is then shaken with the aqueous liquid where the aqueous salts will be present in the aqueous liquid while many impurities remain behind in the organic liquid are separated to get a pure alkaloidal salts. Method 2. The collected powdered material is extracted with water or aqueous alcohol containing dilute acids, and to this chloroform other organic solvents are added, and it shakes well to remove the pigments and other unwanted materials, whereby the free alkaloids are then precipitated by the addition of excess alkalis like sodium bicarbonate or ammonia and separated by filtration or by other extraction of organic solvents. Chemical test for alkaloids. The common chemical tests used to detect the presence of alkaloids are four main reagents like Mayer's, Dragonras, Hager's, and Wagner's reagent. One best example to see about the alkaloid, quinozoline alkaloids is cinchona. Synonym of the bark is Countess bark, Peruvian bark, Jesus bark, Cortex cinchona. Biological source consists of the dried bark of Cinchona officinalis, Cinchona calicea, Cinchona lexerina, and Cinchona sexuruba family of the drug is Rubiaceae. The main geographical source where the plant is being seen is in South America, and it is cultivated in Anaimalai, Nilgiri Hills, and Darjeeling areas of West Bengal. Cultivation preparation. So they are cultivated by seeds. While cultivation, they requires an acidic soil. The rainfall should be from 190 to 500 centimeter. They grow well at an altitude of 1000 to 2000 meters. The seedlings are transplanted twice till they are about half years old. Then they are transplanted at a distance of 1 meter in rich, porous, and well drained soil in slopes or terraces, or at an altitude of 1000 to 2000 meters. And Chincona requires a hot, humid, and tropical climate. The average temperature should be 15 to 25 degrees C, and the rainfall should be 190 to 500 C. Cinchona bark is collected in the rainy season from 6 to 9 years old plant by uprooting method or coppicing method. Since uprooting method, most of the plants are getting diminished, the coppicing method of the bark is being done. And what do you mean by the term coppicing? Means the stem bark near the soil level are being scraped and it is collected from the plant. The barks are collected from the branches, stems, and root by making suitable incisions and are dried under the heat of sun. During drying, the inner surface of the bark, which is quite pale, changes to red color due to changes taking place in the tannins. General characters of the bark. The shape of the bark will be curved quill or double quill. The size of the bark will be 30 centimeter to 1.5 to 2 centimeter in diameter due to longitudinal wrinkles, transverse cracks, fissures, ridges, and protuberances. Gray patch of moss or lichens will be also present in the bark. The inner surface consists of longitudinal lines running from the top to bottom. The fracture of the bark is short in outer bark and fibrous in the inner bark, and the taste of the bark will be very bitter due to the presence of alkaloids. Coming for the root characters of the bark, the shape will be curved or channeled pieces like means of a gutter, and the outer surface consists of conchoidal or mole-like appearances. Lichens and epiphytes will be present in the outer surface. The root bark ranges from a size of about 2 to 7 centimeter long, and the taste will be highly bitter due to the presence of alkaloids. There are 4 types of cinchona, and now we can see the differences of the cinchona species. First species is Cinchona saxaruba, second one is Cinchona calicea, third one is Cinchona legerina, and fourth one is Cinchona officinalis. So coming for Cinchona saxaruba, they range at a size of 20 to 40 millimeter in diameter and 2 to 6 millimeter in thickness. Outer surface, they have well-marked longitudinal wrinkles, relatively few with transverse cracks, and some pieces shows reddish warts. The powders will be reddish brown in color. Coming for Cinchona calicea, the size is 12 to 25 millimeter in diameter, 2 to 5 millimeter thickness, and the outer surface is broad with longitudinal fissures with transverse cracks and about 6 to 12 millimeter apart. The powder color will be cinnamon brown. Cinchona legerina have a size similar to that of the Cinchona calicea. The outer surface consists of cracks which are more numerous and less deep. Some pieces show longitudinal wrinkles and reddish brown warts. The powder will be cinnamon brown in color. Coming for Cinchona officinalis, the size will be 12 millimeter in diameter, 1 millimeter in thickness. The outer surface consists of transverse cracks. They are very numerous and often less than 6 millimeter apart. The powder will be yellowish in color. Microscopy of cinchona. The microscopy of cinchona can be viewed under the microscope by taking the transverse section. The transverse section of the bark shows uniform cork composed of several layers of thin-walled cells containing amorphous reddish brown matter. Below the cork is a reddish cortex composed of tangentially elongated parenchymatous cells with reddish brown and thin walls containing small starch grains. The cortex region, we can see the idioblast containing microcrystals of calcium oxalate, maybe 2 to 6 mu long, and secretion canals are scattered in the cortex. The phloem consists of compressed and collapsed sieve tubes, and the phloem parenchyma are similar to cortex, which are irregularly arranged under large spindle-shaped lignified fibers. The medullary rays are narrow in the cortex region, of 2 to 3 cells wide and almost straight. The longitudinal section of the bark shows brick-shaped cells of medullary rays and longitudinal elongated cells of phloem parenchyma and fibers with conspicuous pits. Constituents. The alkaloids are quinazoline derivatives. Quinine and quinidine contain methoxy groups. Cinchonine and cinchonidine does not contain methoxy groups, which is the main difference between the quinine, quinidine, cinchonine, and cinchonidine alkaloids. Chemical test for alkaloids. Thiophenone test is the confirmatory test for alkaloid quinine. To perform the test, dissolve 1 to 2 milligram of quinine in dilute acetic acid and add 2 ml of water. Then add 2 to 3 drops of bromine water and shake it well. And to this, add a drop of strong ammonia and an emerald green color will be produced. Next chemical test. Cinchona bark powder is moistened with glacial acetic acid and then heated in an ignition tube. Blood red vapors condenses on the sides of the test tube due to the presence of phlobatannins. The third test is the UV fluorescence test. The cinchona bark powder is moist with dilute sulfuric acid, which shows a blue fluorescence in ultraviolet rays due to the presence of methoxy groups. And a small amount of coarse powder, when made alkaline with lime and extracted the alcohol and chloroform, the solvent layer separated out and it is evaporated to dryness to give a residue, and the residue is dissolved in dilute acids and used for the following test. When the extract is treated with the Mayer's reagent, it gives a white precipitate, and the chemical name for Mayer's reagent is potassium mercuric iodide. The next extract is drag and drop reagent. The chemical name is potassium bismuth iodide, which gives an orange red precipitate with the extract. Next test is Wagner's test. The solution of iodine in potassium iodide is called as Wagner's reagent test, and the extract gives a reddish brown precipitate with Wagner's reagent. The last test is Hager's reagent, saturated solution of picric acid. When Hager's reagent is added with the extract, it gives a yellow precipitate for alkaloids. Among all the tests, Mayer's reagent is considered to be the confirmatory test for alkaloids. These are the Mayer's test for the color change. The color first produced in Mayer's test is yellow color. In Wagner's, it will be of reddish brown in color. Next is Hager's test, where a yellow color precipitate is being produced and the drag and drap's reddish brown color is being produced. Uses of cinchona alkaloids. Quinine, which is found to be the major constituent, is used as a best anti-malarial and antipyretic agent, and it is effective against the asexual cells and not against sexual cells gametes, and thus recurrence of malarial attack occurs. Synthetic derivatives now have replaced the quinine alkaloids like chloroquine and primaquine, and anti-malarial drugs. The mosquitoes has become very resistant to synthetic drugs, so now the demand of quinine has increased. Quinine is used as an anti-infective, analgesic, and also used as an oxytocic. Quinidine is used as a cardiac depressant to inhibit the auricular fibrillation. Galenical subcinchona is also used as a bitter toxic and stomachic, but on account of astringent action, the decoction and its infusions are used as gargles. Cinchonidine is used in rheumatism neuralgia and used as an antispasmodic in whooping cough. Substitutes and the allied drugs. The biological source is Remigia pedunculata and Remigia purushiana. Family of the drug is Rubiaceae, and the synonym of the drug is considered to be false cuprea bark. So the color of the bark will be copper red in color. The outer surface will be very dense. The fracture is very hard with the granular and splintery fracture. Though in the microscopy, it contains transversely elongated stone cells which are absent in cinchona. Thereby, we can find out whether the drug is adulterated or not. The Remigia pedunculata contains quinine, quinidine, cinchonine, and cinchonamide. Cinchona lancifolia and the other adulterate drug, which contain abundant sclerids in cortex and medullary rays. The phloem fibers occurs in small groups, and the various species of Cascarilla, Ixostoma, and Stenostoma drugs are also adulterated with cinchona, which do not contain cinchona alkaloids. Thank you for your patience listening.